record. Okay, so they are fastidious. So that means that they need extra nutrients to grow. They won't just grow on that tryptic soy agar that I give you in lab normally. So what we do is we grow them on agar that's got sheep's blood in it. And we buy it. We don't make it here. So we have agar, it's got sheep's blood in it. What turns out, okay, that some of the streps, okay, they have different ways that they actually break down blood. So they have different, they call them hemolysis patterns. Okay, so they name them alpha, beta, and gamma. Whoops, and I don't remember what the symbol for gamma is. I should know that.
I think
okay, so there's alpha, beta, and gamma hemolysis. So essentially, you can look at colonies growing on a plate of sheep's blood agar, and you should be able to see, I'm trying to find, you're going to see different colors. I should open up the file and put it in the, okay, lab resources. So hold on a minute and I'll move this file over so you can see the pictures. Where's my staph and strep stuff? Oh, wonder what I did with it. All right, so let's just do I have pictures of it in the lecture?
I think so. I can pull the PowerPoint up.
Yeah, here, I have it. I have it right here. So skin.
On slide 15.
Okay. Oops, here, Alicia's here. Hey, Alicia. Oops, she's not in there yet.
Hello.
Hey, how are you?
I'm good. I'm good. How are you?
I'm good.
That's a good topic. I was gonna ask about this, alpha, beta, and gamma.
Oh, good. Yep.
Yeah, I just walked in home from work, so sorry.
It's fine. It's cool. I was actually had it open right at the right time.
Oh, thank you.
Yeah, let me try to put this file, this PowerPoint file in.
Kristen there?
Yeah.
Hi, Kristen. I don't know you, but I feel like I know you.
Yeah.
All right, I'm going to try to put this file on my iPad so I can open it on here, hopefully. Okay, OneDrive. Oh, shoot.
I missed you guys last week. I tried to log in and it just kind of kicked me out.
I know, nobody came, so I don't know if that's what was going on.
Oh, okay.
Yeah. I had to work last week. That's why I wasn't there.
Okay. Well, then I don't know what was going on.
That's okay. We're here today.
Yeah. Okay, so let me open this up from, so if I can import, and, oh, I have skin. I don't know what I was, no. Where's it at? Skin diseases, why isn't it coming up? Import. I need somebody to give me a class in file management. Micro Spring 2020, what's this? Microbiology, micro lab activity. Gram negative. Where's skin? Staph and strep, here. Here. Oh, come on.
How are you, Kristen? Did you start the new chapter yet? What was it? I'm on 1718, but this week chapter. What is the chapter this week? I don't even.
This week is the skin diseases.
Yeah, I haven't started. Have you started that one yet?
Yeah, I just started it, but then I got to this part and I got confused.
Shoot, I'm trying to get this stupid thing to load. All right, it's okay. Let me just new. I'm supposed to be able to import new.
When is the Elisa from lab?
Eliza is how they
Liza.
say it.
Okay. When is that due?
I think I have it due not till that next week.
Okay, 30.
Yeah.
Because I'm
And
gonna do everything. Yeah.
I think it's interesting. I don't know. You guys might think it's really boring, but it goes with the immune system, and it goes with the immune system. It goes with this. It goes with infectious disease detection. So it goes with a bunch of stuff that we're doing right now. I'm getting
This
frustrated. Okay, so let
be
me
on
just.
exam? Is that that's not part of the exam, right?
No. So I'll have the all that ELISA stuff.
Yes.
No, that we'll put that on the lab test, and the
Okay.
lab test is open
Yeah.
book, so it'll
Okay. I
be.
just want to mention. Okay, sorry.
No, it's fine. Go ahead. What?
No, I was just doing. There's a lot coming up, so I'm doing what like minimum before the exams. I can study for the exam more. That's why I'm asking.
I understand. It says importing the document, but it's not bringing it in. There it is. Okay. So do you guys see this thing that says staph and strep now?
Yes.
Okay. So staph and strep, we end up talking about this in this chapter, even though it's not in this chapter in the book, and it goes with what's going on in lab. So staph and strep are two organisms, and they can cause infection all over the body. Okay, so they could be on the skin. Okay, so staph is on the skin all the time, but they can be on the skin, they can cause skin infection, they can cause infection pretty much anywhere. Okay, now, do you have to, they're both gram positive, right? So they're gram positive. One is going to be chains, and one is going to be clumps, right? Clusters. So that was just a little reminder I have on here. Okay, so usually what we're going to do, okay, is in the book, I talk about the blood and the hemolysis patterns. Okay, so strep is hard to grow. It's fastidious. So it doesn't like to grow on any media that we have. So we have to add extra nutrients to it to get it to grow, and that's blood agar. So we add 5% sheep's blood to it, which is kind of gross, but it just looks like bright red. Okay, so it has to have extra nutrients. So let me go over here and let me show you the hemolysis. Okay, so there's 3 different kinds of hemolysis that they use. So there's alpha, and I'll post this. Okay, there's beta and there's gamma. Okay, now all it is the different organisms break down the hemoglobin in the blood different ways. Okay, so some of them break it down completely, some of them don't break it down, and some of them break it down a little bit. Okay, so if you look, alpha looks kind of green, and it looks like it's not a pretty green, right? It's like an old blood green. Okay, beta, it totally breaks it down, so it's clear around the cells. And then gamma is there's no hemolysis. So if you look, hemolysis is hemo and lysis. So literally, it's just the breakdown of the hemoglobin in the blood. Okay, so some of them can totally break it down. Some of them break it down partially, and some of them don't break it down at all. And it's cool that you can actually classify them based on this. Okay, so if they do a swab of somebody, they'll put it on, like if you go get a strep test, right? They think you have strep throat. So they'll take the 2 swabs, and they're gonna rub them in the back of your throat, right, and make you gag. And then they'll use one for a rapid strep test, and which is an antibody-based test. And then the other, they're gonna plate. They're gonna put it on the plate. So then the different kinds of strep that you see. So one is Streptococcus pneumonia, and it looks really sticky and goopy, and it is green. The colonies will look kind of green around it. So it's like a real kind of disgusting green. It's not pretty, right? So there's some of it there, how it looks really, it's not pretty green, right?
Yeah.
And then beta, it's totally clear. Okay, then that one is actually, that is Streptococcus pyogenes, which that's the organism that causes strep throat. Okay, and then there's gamma. And the gamma ones, they're not as pathogenic, and they don't break it down at all. Does that make sense?
Yeah.
Yeah.
Yeah. Are you going to keep recording the rest of the questions? Because I actually have to go, but I was just curious if it'll still go up.
Yep. I will
Okay.
do that. Yep.
Thank you.
Yep. Bye.
See you on Wednesday.
Alicia, what question? Do you have questions?
Going back to host defenses because I'm not on this chapter yet. And when I said alpha, beta, and gamma, I was thinking about the molecular defense.
Okay.
There's a table of properties of type 1 and type 2 human interferon.
So that's,
do we like, can you go over these alpha, beta, and gamma? Because I didn't understand those.
oh, about the stuff for the skin? Or do you want me to go over?
That's one of the slides from the last chapter.
Okay.
Is molecular defense.
Okay.
And, let's see.
So the
Therapeutic.
molecular defenses.
Yes.
Okay.
Interferon, and there are steps that I understand that, and then there in the bottom it says there's a table 17.3.
Okay.
Property of type 1 and type 2 interference, and I don't know.
Oh, you don't have to know the difference between
No.
type 1 and type 2 interferons.
Oh, okay. Yeah, on this on the slide, on the video, YouTube video, I don't think he talked much.
No, just know in general what interferons do. So they're released in response to a virus, right?
Yes, yes.
And they're kind of kamikaze like. So a cell that gets exposed to a virus will start to make interferon, and then it'll trigger the other cells to make interferon. So it stimulates the production of more interferon. But you don't have to, I don't spend too much on the details in here. It's just really general.
Okay. And what about 5 types of white blood cells?
Oh,
Much more than 5. And I was wondering, it says one of the questions was,
right.
what are the 5 types?
Okay,
The
so
last 2 I wasn't sure about which one to pick.
yeah, in the book she puts more of them. In anatomy we only usually talk about. Okay, so hold on, let me try to get this open. Come on.
Got a basophils, neutrophils.
Yeah. So I usually think about, so the never let monkeys eat bananas.
Oh,
Yeah. Oops, can you see that?
yes.
Okay. So never let monkeys eat bananas. So neutrophils.
Oh.
Okay, lymphocytes. Monocytes. Eosinophils. And basophils. Okay, now they add in dendritic cells in our book.
Yes.
And I don't know why, like they don't usually talk about those in the anatomy book. So they're phagocytic. So that means they do phagocytosis where they gobble everything up, right? And I always think of them as being in the skin, right? So they're in the skin. They I don't know why she keeps them. I don't know. Like different places list how many we actually have. So just go with this for right now. So never, so I always say never let monkeys eat bananas.
Yeah, that's easy to remember. And
Yeah,
as far as basophils, I guess it says what do they do, and when would they be elevated? So what do they do? I know from
right.
rocks, but when would they be elevated? What would that be?
Okay, so like basophils is going to be if you have an allergic reaction to something.
Okay.
So basophils are if you have an allergic reaction. Neutrophils are gonna they're kind of generic, so they go up anytime there is an infection. So lymphocytes are going to make antibodies. Oh, you already know those. So you want to know when they're going to increase. Okay, so eosinophils go up if you have a parasitic infection.
Okay. How about allergic response too?
And allergic too, yep. Yep. And then monocytes go up when there's a viral infection. And
Yeah.
then
Lymphocyte.
I know. What do I have for lymphocytes? Do I have anything about them?
Lymphocytes produce antibodies necessary for immunity. But
Yeah, so they don't really, so they're gonna, it's not like a certain pathogen is gonna trigger them. But anytime there's an immune response, they're gonna be in there, they're gonna start making antibodies.
okay.
Yep.
Okay, that's great. So make antibodies.
Okay, now all of these, right, can be phagocytic. So the neutrophils, the lymphocytes, the monocytes, the eosinophils, they can all, like the cell can gobble up another cell. The only one that can't do that is basophils. Okay, so basophils are different. And what they do is they'll release histamine. And histamine makes capillaries leaky. So it makes, like when you have a cold or you have an allergic reaction and your nose starts running and your eyes start watering, that's histamine being released.
Okay. Oh, what else I want? The role of phagocytic white blood cells, would that be just to find, adhere, ingest, and digest microorganisms?
Yep. Yep.
And the chronic inflammation, we have syphilis, leprosy. Do we need to just know generally what are kind of chronic inflammations, or more what causes it as well?
I think just in general what they are.
Okay. Okay.
Yeah. And some things they just don't ever heal.
What would be artificially acquired passive?
Okay, so that would be, so artificially acquired passive.
Because you put like antitoxin,
Yep.
antivenom. I don't know what antivenom.
Okay, so if somebody gets bit by a snake or a poisonous spider or something, they have, and so it releases a toxin. So the toxin might look like this, right? They have antibodies that they've designed, so anti-venom or antitoxin, and it'll bind to the toxin. So instead of the person making the antibodies, we are giving it to them. And it's passive because they're not making them, and it's artificially acquired. They're not, it's just totally passive. We're giving them an antibody. that'll bind to
It's
something.
always related to animals?
No. So this is what's interesting. Okay. So some vaccines, they call them vaccines, right? They call them vaccines, but they make antibodies against some viruses.
Oh,
So they make these things, they're called monoclonal antibodies.
yeah.
And so MAB is how they abbreviate it. And you see, if you're looking at a magazine, they advertise them for like rheumatoid arthritis and plaque psoriasis and things like that. So basically, and one good example, there's this virus it affects babies, premature babies. So it's RSV, so respiratory syncytial virus. So it's around all the time, and adults can get it, and it's just like a cold. But a premature baby, it makes them really sick, and they can end up in the ICU. So what they've developed, and there's no vaccine. They don't have a vaccine that they can give them. So they call this a vaccine, but it's not really a vaccine. What they do is they have these monoclonal antibodies that have been designed against part of this virus, and they give them to the kids. They last maybe like, I don't know, I think they dose them every month or so over cold and flu season. So they're not making antibodies, they're giving them the antibodies, and they don't keep making them, so it goes away over time.
Interesting.
So it's kind of funky because they call it a vaccine, but it's not really a vaccine because you're not making any memory to it.
Yeah.
But they make these in Frederick at AstraZeneca.
Oh, really?
Yeah. Yep. They make like the world supply of it.
I did not know that.
It's called it's called Synagis. Synagis. It's SYNAGIS.
Synergist. Okay. Oh, this is a tough one for me.
What?
Immunoglobulins.
Oh,
I
think they're tough.
Something good to know, I guess, in life generally, because sometimes when I do a blood test, you see this, and I don't know.
I know, and I think it's hard for everybody, honestly. So I want you to understand, okay, that there's more than, like, you don't have to know everything that's in the book. So what I want you to know, and I have on the worksheet, I have information for you guys to put in the properties of these immunoglobulins.
Right, that's what I'm looking right now. I try to do it. I would summarize, like, what's the easiest way to remember, or what's the most important about those IgGs and AMR?
So the biggest one that is most prevalent is IgG, right? Yeah. So IgG is the most important one.
80%.
Right? So that's, and if I ask questions, I talk about that one, and that's the one you hear about all the time, right? So I don't expect you guys to memorize that table, and I know I have questions about it on there, but I just want you to know, okay, antibodies are, they're made out of protein. Yeah. They have that typical Y shape, right? They have that little Y shape. Most of the Y is common to all antibodies, but there are parts of it, like the small piece, that it changes between organisms, right? So depending on what you're reacting against, that part is going to change. Okay. I know, I don't, I go back and forth because I don't necessarily think you need to know all this. It's cool.
Yeah, I was trying to fill this table there.
Yeah. I don't usually ask that many questions about it. Just know about IgG. So
okay.
It's the main class of antibodies.
Yeah, I like to understand what I'm, not just memorize it, but actually know what
Yeah, and it's if you have to use these, like you
Mhm.
take a whole class on
Really?
immunology. So you
Wow.
have like, right, 15 weeks of these two chapters. So we're kind of just like, okay, here it is, go,
And
right?
then
And
it talks
so I
about
don't,
AGM and the blood types, and I just stopped right there.
yeah,
'Cause,
so don't
okay,
worry about it. This just know in general.
okay.
Yep,
And IgG is the
is the
most
main
popular.
one. I know.
Okay.
Sometimes I'm like,
Thank
oh, you got it, but I don't think you if you need to know them in more detail, you're going to learn them again.
Yeah, I was excited for a minute because oh, we see it in life on a blood test. At least I do. But
Right.
then
It's
I don't.
it's too much for like a lecture.
Right. Okay. And last, this was the last question on the worksheet. Primary response versus secondary response.
So primary response is the first time you see a pathogen, right? So it's the first time. So it's going to take a while to get the antibodies made. Okay, but then the second time you see it, it's hopefully it doesn't take as long and you get more antibodies faster. So you get exposed to it again. And hopefully you make more antibodies before you start to actually get sick.
Okay, because the memory cells
Yep,
already.
because of the memory cells. So memory cells just have to be turned on, and then they hopefully can start pumping out antibodies so that you don't, that you, so they start making IgG faster. So
Okay.
you don't get sick.
Okay.
I know there's a ton of stuff in here, and I don't go very far into it at all.
Yes, I try to read from the book, study from the book, but it's really overwhelming sometimes
Yeah,
because you can just read about 2 pages about something and then never use it actually.
they put everything but the kitchen sink in here.
Yeah.
There's
And as far as interferon described mechanism, your description was pretty much easy and to understand, like
yeah.
one sentence. But in the book, there are 7 steps.
Yeah, you don't have to know all those 7 steps. No,
And you know
just
what? I
know.
didn't summarize it all nicely, but I wrote
Yeah,
it down to 7.
yeah. Don't worry about all 7 steps. And I don't. Yeah, no,
Okay.
don't
Um,
worry about.
as far as the steps, I think inflammation, right? We should know that one.
Right,
The
just know the general. Yep.
yeah. Let me see what else do I.
I don't even see it. Where is it in here? You said it's in 17.
I have it written down. Let me read it for you.
It's
Virus,
okay.
virus infects the cell, signal sent to the host, the nucleus. Host 3, host cell gene sends the signal, I guess, no, activates the interferon. 4, they synthesize and being synthesized and released. 5, bind to neighboring cells. 6, signal sent to nucleus again. And 7, cells stimulated to produce antiviral. I don't know. I just look at it. I was like, wow, this is so
Yeah, see. Okay, there it is. Okay, yeah, I see it now. Okay, there's. Oh, yeah.
wow.
I just want you to know that it's against a virus, right? And
Okay.
sometimes, and they use them in cancer treatment. So they found that they're successful against, attacking cancer cells. So
Wow. It's promising the vaccine against the
yeah,
virus.
right. So there's 2 of them that they think are good. So
Do you think the second one is a little bit better?
I don't know. I have to read about it. I haven't read about it yet.
95% on.
Yeah, it's 95%. And I need to look and see what kind of vaccine. The first one is mRNA, and I don't know that we have any mRNA vaccines available in the world. So
Wow. That's Whoever
But
takes
if it works, it's cool.
yeah. I'm going through to see if there's anything else that I need.
Okay.
I have a question about. Antigen antibody reactions. Well, I have a question mark here around this slide. Agglutination component
Oh,
normalization. One of the slides, slide 20 of the PowerPoint.
okay. So let me see.
That's again IgA, IgG, IgM. I don't know.
So I think what I was trying to get you guys to see, hold on a minute though. I don't have that PowerPoint open.
Oh,
Hold on. So immune system. Yeah, all this craziness. In the old days, right? I could just talk to you. See you guys in class.
and the other. Oh yeah, what happened to the other class? We won't be able to.
So they're closing on Monday and Tuesday after Thanksgiving.
So we can't start our lab.
So I don't know how we're gonna actually finish it all. So you guys will meet next week, right? You come this week.
Yeah, yeah. Can we do like both for 2 weeks in a row? Like one longer one, maybe? I don't know.
I know. I have to come up with a plan. I'll talk.
Yeah.
I was thinking what I would do is I would meet with you guys next week. And then the week after that gets messed up, right?
Yeah.
So you guys, everybody ends up with like a day.
Right, and we can't start and finish on the same day, right? Because you have to go.
No, there's not enough time to start and finish everything on the same day. So I have a virtual thing I guess I'll do, and it's on Staff and Strap.
Okay.
So I'll just I'll make a video to go with it. Last year I did it. It was a virtual lab.
Oh
So
yeah.
yeah, it'll be all right. It's not the best, but I don't know.
Well, we still got lucky to meet most of the time.
We did, yeah, I was right.
I didn't believe we are gonna last that long, but that's great.
I know when we first said we were going to do it, I thought it was not going to make it, right?
Yeah, that's what I thought. I was telling my boss to, if he allow me to leave early because I wanted to take this class, and he said, "Okay, go ahead. I have a feeling that after 1 month, everything will be virtual."
Uh,
No, nothing happened.
I know so far we've made it.
Yeah,
But
that's awesome. It's made a lot easier in person.
yeah, right. Just to check in with everybody. It makes it easier on me too, right?
Yeah.
Okay, so you're the agglutination.
Yeah. So this was the last question I have.
Yeah, okay. So agglutination is when, so there's a couple different things that happen with the antibodies. Okay, so the antibodies, they look like, right? They look like these little Ys. And this is the part where they actually can capture things, right? So if they capture it, they can do different things can happen in your body to get rid of the antigen, right? So the antigen's what's causing the allergic reaction, and then the antibody,
That's what's fighting.
yeah, is what's detecting what we're making towards it. So once the antibody binds to the antigen, different things can happen. So one thing that can happen, and I have complement mediated effects. So basically what that means is we can kind of tag it. It gets tagged with the antibody, and then the macrophages will come in and they can break it up, ingest it, phagocytize it. So it tags it. So it's not like it's just wandering around looking for something random. It's actually marked. They can also kind of neutralize the antigen and bind more than one of them together. So it kind of clumps them together. And when we clump them together, that's called agglutination. So clumps antigens. So agglutination. And then neutralization is we just take it out. so that it can't infect anybody. So or cause disease. So that's what they're doing with the kids in the monoclonal antibody. So it binds to it, and then it can't make them sick, and it triggers their body to attack it. So the white blood cells that they have can engulf it. The monocytes can break it down.
Oh, okay. So it either marks it for ingestion
Yep,
or creates the clumps.
yep. And then
Or,
the clumps can be digested.
right. And then the same with neutralization.
Yep, neutralization means it just kind of makes it inactive
Okay.
so that it can't cause an infection.
Oh, that was helpful. Thank you.
Yeah, yep,
Thank you. I appreciate your time.
yep. That's no big deal. So now tomorrow you're coming at 10?
Yes, please.
Okay.
Thank you so much. I will, this one time I had no choice in terms of.
It's all right. The 10 o'clock class is small, so it's,
Oh, I was wondering if there's enough people, but I said, well, her to ask. So thank you for that. I
yeah, the 10 o'clock doesn't have very many people in it. And
Oh.
gradually I've been losing people, so it's, it all works out.
will. Okay.
Okay.
Yep.
All right. We'll see you tomorrow.
See you tomorrow. Bye-bye.
Okay.